Okay, perfect. So, after having mastered purine and pyrimidine synthesis, let's now talk about the DNA replication. Okay, again, we just need 1 hour of attention, and you should be knowing everything about DNA replication and how the board will test you. Okay, so keep writing and stop me right there, and then if you have any questions. So DNA replication. So, first thing first, if I say we are replicating the DNA, we should know at which phase of the cell cycle we are doing this, right? Because if we are replicating the DNA, the point of DNA replication, we have 23 pairs of chromosome from mom, 23 from dad. At end of the day, we want to divide that cell into two parts. So how will we do that? Okay. So this should be replicated. 23 should be replicated into another 23, and this 23 should be replicated into another 23, and you should be finally making two cells with 23 pairs of chromosome each. So this is where the DNA replication comes. And if I talk about the cell cycle, we have the gap 1, then S phase, the gap 2, and then finally we have the mitosis. Question: in which phase of the cell cycle we are doing DNA replication? And your answer is
S phase.
wonderful. What a start of the day. Excellent. So that's your S phase of the cell cycle where the DNA replication happens. Okay. Now, after knowing in which phase of the cell cycle DNA replication happens, let me just go back to the basics. We talked about nucleotides. DNA is nothing but two strands of the nucleotide. Okay. These are the two strands of the nucleotide, right? And if the way of writing the DNA is one strand has to be 5 dash, okay, 5 dash to 3 dash, and the other strand has to be complementary, which is anti-parallel. Okay, so if this is 5 dash to 3 dash, the other strand has to be 3 dash to 5 dash, right? Now tell me, what is 5 dash end? Is it the phosphate or is it the hydroxyl group? Somewhere we discussed
Phosphate.
very good, Bobita. How do you know this?
Sir, phosphate is down, so 5 is down. Sorry, phosphate is up, so P means Pella. So I'm
Very
remembering.
good. Impressive. So what is 3 dash?
It's OH.
Very good. So this is your hydroxyl group, which is the 3 dash end. The 5 dash is the phosphate. And if you also have hydroxyl group here, then this is called ribonucleotide. If you don't have hydroxyl group here, we call it deoxyribonucleotide. Okay. And of course, as the place of one, we have the nitrogenous base. Okay. So this is your nucleotide. We have the nitrogenous base, we have the sugar, and we have the phosphate, and this is going to make your nucleotide. Now the incoming phosphate has to attach to what part? If I say I have the another nucleotide, the phosphate of the incoming nucleotide will attach where? So this is the new
3
phosphate.
dash.
Very good. So it is going to attach to the 3 dash. Uh, so this is your nitrogenous base. So this phosphate is going to attach to this 3 dash. And what kind of bond do we call this? What is this bond called as? 3 dash to 5 dash phosphodiesterase bond. Okay. This is 3 dash to 5 dash phosphodiesterase bond. Now, so I want you to notice that what we are going to do is we are replicating the DNA from 5 dash end to 3 dash, because every time the phosphate of the incoming nucleotide will come, that is going to attach to the 3 dash of the 3 dash end of the previous nucleotide. So we are going to replicate from 5 dash to 3 dash. Okay, know this fact. So I'm going to write this down, a very important thing, one fact about DNA replication. It is going to replicate in 5 dash to 3 dash direction. Okay, now if I say if it's going to replicate in 5 dash to 3 dash direction, in what direction it has to read the parent strand? What's your answer?
3 dash to 5 dash.
Yes, very good. So it will read from 3 dash to 5 dash and it will replicate from 5 dash to 3 dash. Now you know what is 5 dash. Now you know what is 3 dash. Another way of explaining this is a strand which you read from 5 dash to 3 dash is called as the sense strand. Okay. And the strand which you are reading from 3 dash to 5 dash is called as anti sense strand. Okay. Now, please remember, when I will talk about genetic code, this sense strand is also called as coding strand. Okay, do not confuse this. Okay, the sense strand is also called as coding strand, and this will be exactly equal to the codons of the RNA, except instead of T, you will have U. Okay, just register this in your mind. If I say ATG, this is from 5 dash to 3 dash. This is very important. This is 5 dash to 3 dash, from direction of 5 dash to 3 dash. If I'm reading ATG, this is the coding strand of the DNA. Okay, now if I want to write down something complementary from 3 dash to 5 dash, what should I write here? What should be other antisense strand of the DNA sequence should be?
GTA.
GTA. So what's the complementary of A? I should write T here. A. Is it correct?
Yes, sir.
So how should I read this? TAC or CAT? We should always read from 5 dash to 3 dash. Okay. So in your exam, if they give you option of ATG, CAT, and if they don't write 5 dash to 3 dash, just presume that it's 5 dash to 3 dash unless until mentioned. Okay. So always read the DNA from 5 dash to 3 dash. Okay. And this is going to be your complementary strand. Okay. Now, if I say this is your complementary strand, this is your coding strand. In other words, I can say this is your sense strand, this is your anti-sense. Nonsense. We don't call it nonsense, but we do have some nonsense mutations. This is sense, this is anti-sense. Okay. Um, if I want to write down codon of RNA, it is going to be exactly similar to 5 dash to 3 dash. In other words, just note this thing, everything down. I'm just going to switch to the next page. I just wrote ATG from 5 dash to 3 dash, and its complementary sequence would be this one, C, A, and T, right? CAT. Now, if I want to make RNA, this RNA or the codon of the RNA will be exactly similar to this coding strand. Okay, because the way I'll make RNA will be complementary to this one, right? The way I'll make RNA is going to read this. That's why it's RNA. When you're making your RNA or you're doing transcription, it's going to read this strand, which is also called as template. Okay, now you're saying, hey, what is this? What the hell is happening? So I told this is coding strand, which is also called as sense strand. The template strand is also called as non-coding strand, which is also called as uh, antisense. Okay, and this is all similar terms which reads from 3 dash to 5 dash. Now remember, if I say this is template, if I want to make RNA, the RNA polymerase is going to read this strand, right? So it is going to make a complementary strand to this one. So it has to be 5 dash here and it has to be 3 dash because we are making complementary to this strand. Now tell me what should be the complementary strand sequence of RNA should be. What should I write here starting from 5 dash?
U T G.
AUG. That's very correct. Now notice this. You don't need to do all this mess. Okay. If you just remember, the coding strand is equal to the codon of the RNA, except you just have to move from T to U. Now the tRNA will come. The tRNA will read this codon, okay, and it will bring one amino acid with it, and that's how it is going to do a chain of amino acid. And what amino acid do you think AUG codes for? Methionine, right? That's our
Methionine.
very good. That's methionine. That's our first amino acid. Okay. So right now you just need to remember the coding strand of the DNA will be exactly similar to the codons of the RNA. If they talk about genetic codes and they write 3 3 sequence, that means you have to assume that if it's T somewhere in the coding strand, that means it's the coding strand of the DNA. If it's U somewhere in the coding strand, that means it's the coding strand of the RNA. It has to be exactly similar. Instead of just T, you have U. Any questions on this one? Crystal clear? Okay. Now, I just told you that we are going to replicate the DNA from 5 dash to 3 dash, and we are going to read the DNA from 3 dash to 5 dash. The mode of duplication or replication of the DNA happens semi-conservatively. What is semi-conservative? What is semi-conservative? So if I say this is the blue DNA, which is also called as the parent DNA, when it replicates, it's going to separate into two strands. Okay, let's say this is the one strand and this is the another strand. These are the old one, old. Okay, now I'm going to make one new strand which is complementary to this old. So when you make a new DNA, one strand is old and the other strand is new. Okay, this phenomena is called as replication with semi-conservative phenomena. That means you are conservating at least half of the parent strand, okay, and you are making one new complementary strand. Okay, so each DNA will have one parent strand and it will make one new strand. This phenomena of duplication or replication of the DNA is called a semi-conservative mode of replication. There are some proteins which do not multiply semi-conservatively. An example of that proteins is which multiply non-conservatively. They are histones. Okay, histones, meaning let's say this is your cell and this cell has 3 histones. When it duplicates, all these 3 histones will go to one cell. The other cell will have entire new set of 3 histones. Okay, it's just like when a couple is getting divorced, one will take everything, the other has to do everything new. That's a bad example. So once one cell will get everything, the other cell will have to make entirely thing new. This is how the histones replicate non-conservatively. That means one has everything new, the other has everything old. Semi-conservative is one each will have at least one strand old, and it has to make one strand new. Okay, so that's your semi-conservative mode of replication. Now let's get to the fun part of the DNA replication. um, First thing I'll talk about is origin of replication. Okay, so keep writing down the headings and we'll exactly talk about the steps of DNA replication. So first thing is origin of replication. Origin of replication. Okay, also called as ORIC. Origin of replication. Now, if I want to replicate my DNA, it has to find some protein has to find at least one site where you can start the replication. Okay, let's say this is your DNA. Where should I start the replication from? The answer is from the origin of replication. Okay, now there is one special thing about origin of replication. You have to find such a spot in DNA which is very easy to separate. Now you tell me, what should be easy to separate? It should be AT rich sequence or GC rich sequence.
AT rich.
Wonderful. That's AT rich sequence, and we call it AT, right? This is also called as Tata box. Okay, Tata box, the setup box. That's the origin of replication. This is also sometimes called as Pribno box. Okay, we have different origin of replication: Tata box, Pribno box. Okay, so let me just write down Tata box here. Okay, so that has to be AT rich sequence, and it's easy to decode this because A and T will just have double bond, G and C will have triple hydrogen bond. So AT is very easy to separate as compared to GC. So origin of replication has to be AT rich. Another point here, since we have 2 meter length of DNA, if you just open up one DNA, one chromosome, it has 2 meters of length. We have to replicate that DNA very fast, right? Now we cannot afford to have just one origin of replication. uh this is a point where you separate and you just keep on multiplying. You run for 2 meter, and then again in other DNA you find one spot. We don't do that. We have a way by which we can replicate very rapidly. What should be that way? What do you think? How can we replicate our DNA very rapidly? By having multiple origin of replication. Okay, and that's your question. So 2 points from origin of replication. It has to be AT rich. The second point is it has to have, if eukaryotics wants to multiply rapidly, and the way by which you can make it less time consuming is by having multiple origin of replication. Okay, these are all your questions. Okay, prokaryotes may have just one origin of replication. Eukaryotes may have multiple origin of replication because of their longer DNA. Perfect. Now, the DNA replication, I said you find out the origin of replication. It's actually bidirectional. In exams and books, we have this famous diagram where it just shows one way, right? This is the direction where you are replicating. In reality, is actually all bidirectional. Bidirectional in the sense, this is your replication fork, right? This is y shaped on both the side. This is your replication fork. When you open up the DNA, the bubble which it forms, it's called the replication fork. Actually, the replication is bidirectional, not just one way. Okay, and one will be leading strand, the another will be lagging strand. Okay, that's your origin of replication. Now, once you find out that AT rich sequence, you open up and you make this something called as replication bubble or replication fork. Okay, so your second point in DNA replication, you open up from the origin of replication, and you form this Y-shaped fork curve on the two ends, which is called as replication fork. Okay, one side will have leading strand, the other side will have lagging strand. That's your replication fork. Okay, now then third thing is you find out this origin of um, replication. You want to separate those DNA. There should be one enzyme which keeps on separating the two strands of the DNA. Do you know the name of that enzyme by any chance? One enzyme which should constantly go in this direction, separating the two strands of the DNA. What is that enzyme?
Helicase.
Helicase. Very good. Helicase. So I'm just, from now onwards, I'll just draw one replication fork. So one enzyme is here, which is called as helicase. What's the function of helicase? It will open up the both strands of the DNA, and it will go and unwind the DNA. One thing you want to remember, if your duplicated strand is moving in the direction of helicase, that's called as leading strand. If your duplicated strand is moving away from the helicase or your replication fork, that's called as lagging strand. Okay, that's your leading strand and lagging strand. There is one disease where we have problem with helicase. What is that disease? one disease where we have some problem with helicase, or we have deficiency of that gene which can fail to produce helicase.
Dermatomyositis.
No. Starts from
There's something balloon something as a
Bloom
syndrome.
syndrome.
Bloom
Bloom
syndrome.
syndrome. Yeah.
Bloom syndrome. So that's your first thing and clinical vignette which you want to remember. And remember, this helicase is energy dependent. It needs energy to separate two forks of the DNA. So if you have Bloom or BLM gene mutation, you will not be able to make this helicase protein, an enzyme. So you won't be able to separate the two strands of the DNA, and you end up getting a syndrome which is called as Bloom syndrome. Now, since this is an enzyme, you would say, since this is an enzyme helicase, the mode of inheritance has to be autosomal recessive, right? Enzyme has to be autosomal recessive. And ultimately, if you are not able to separate the two strands of the DNA, ultimately it will lead to chromosomal instability, right? It will lead to chromosomal instability. And if your chromosomes are not working, you won't be able to duplicate your cells properly. So you'll have problem in the growth, right? So patients will have growth retardation, right? You cannot be able to grow your facial bones, so facial anomalies, some skin rash. And since you're having problem with duplication of everything, you'll have immunodeficiencies as well. Okay. Bloom syndrome, growth retardation, some rash, facial anomalies, and immunodeficiency. I'm going to write down immunodeficiency because that's important. Okay. Immunodeficiency. Now, that's your helicase. Now let's move forward. We have separated the DNA, okay? So we found out the origin of replication, which is AT rich. We want to separate the two strands of the DNA with the help of helicase. The helicase is deficient in Bloom syndrome. Now we separated the DNA. Now they are like very, very early boyfriends and girlfriends, okay? Early boyfriends and girlfriends. Not for very long time. Not even married. Not married for 10 years. Very, very early. So if they're very early, do you think they would like to be separate? No. Early boyfriends and girlfriends don't like to be separate, right? They just stick together. So when you separate that, uh, the single strands of the DNA are very, very excited to come close together. Okay. They'll find every spot to come close together. That's one problem. So every new boyfriend and girlfriend will have their mom who will know everything. Something is going on. So the mom will come. They'll not allow these boyfriends and girlfriends to be together. So our, what was the number? Fourth, fourth protein is called a single stranded binding protein. Okay. What it will do, it will keep the two strands of the DNA separate. So single stranded binding protein will bind with the one strand here, and single stranded binding protein will bind with the another strand here, SSB. This is how it will keep the two strands of the DNA apart. Okay. The reason why you need single stranded binding protein, one, is the DNA has very high tendency of re-annealing. Okay. Re-annealing is when your DNA, just like boyfriends and want to come together. The second one is when you are alone, you are very, very vulnerable for attacks, right? Same like single stranded DNA. When you are alone, this single stranded DNA is very vulnerable to attack. Okay, very vulnerable to attack. So it's just going to digest this part of the DNA. Okay, so if you are alone, it's vulnerable to attack by nuclease. Okay, so it's just going to come and digest. So to prevent the digestion of the single strand and to prevent two strands of the DNA to reattach, we have this protein which is called a single stranded binding protein, which will keep the two strands of the DNA separate. Now, fifth one, if you are using your helicase, you are opening two strands of the DNA When you're opening your two strands of the DNA, what will happen to these? You are having helicase. You keep on opening. Finally, this twisted part will get more twisted. It's just like you are having two ropes. You are trying to open it up. The end part will get more twisted, right? And it has a tendency of forming supercoil. So you form supercoil like this if you are trying to separate two twisted things. Okay, so if you use your helicase, you open it up, it's going to form supercoils here. Okay, those are called as supercoils. Now we have an enzyme, or we have one special enzyme which can come, we have one special protein which can come and reduce these supercoils. Does anybody know the name of this one?
Topoisomerase.
Topoisomerase. Perfect. Topoisomerase. Okay. Now we have um, different kinds of topoisomerase: topoisomerase 1 and topoisomerase 2. Okay. Topoisomerase 1 is designed in such a way, let's say this is your topoisomerase. It is designed in such a way that it can cut one strand of the DNA, it will unwind, and again it will join with the help of some glue. Okay. Topoisomerase 2 has two cutters. It can cut both the strand, put just one nick here, cut both the strand, unwind them, and again you rejoin. Okay. So the main function of topoisomerase is cutting the DNA, you unwind it, and you again rejoin. Now we have topoisomerase in prokaryotes and eukaryotes. Okay. Now the topoisomerase which we have in eukaryotes. So eukaryotes have, so we have topoisomerase 1, 2, and 4. Okay. These two are present in eukaryotes and these two are present in prokaryotes. So topoisomerase 2 is kind of common. Okay. Now, the reason why this is important, because we have drugs which can act against topoisomerase, and that's kind of very interesting, that if you give some drugs, which can inhibit topoisomerase, meaning this part of the cutter of the topoisomerase is called as nuclease. It means it's just going to cut down the DNA. If you give this drug, it will keep on cutting the DNA, but it will never allow to join. So DNA will keep on accumulating the supercoils and the DNA will fracture. That's the mechanism of the drugs which can inhibit the topoisomerase. Okay. Now we have different categories of the drug. We have the drugs which can block topoisomerase 1, and we have a mnemonic for that. What's that? Name of two drugs which can inhibit topoisomerase 1. Can. I can. Irino. T can. Okay. I can. So remember, I can. I is one, can. So whenever we have irino, tican, topo, tican, oops, irinotican, topotican, that's topoisomerase 1 inhibitor. Okay. Now we also have some topoisomerase 2 inhibitor. Remember, we have coin has two sides, right? Coin has two sides. So the drugs has to be sides. Any guesses? Etopo side and teniposide, right? Etoposide and teniposide. Irinotecan, topotecan can block topoisomerase 1. Etoposide, teniposide can block topoisomerase 2. Finally, we have a drug which can we have a drug which can block prokaryotic topoisomerase, which is also called as DNA gyrase. What is that drug? Topoisomerase is the other name of topoisomerase.
Fluoroquinolone.
Excellent. So the drugs which can block 2 and 4 in prokaryotes is fluoroquinolones. Levofloxacin, moxifloxacin, all floxacins. Okay. So fluoroquinolones, they will ask you the mechanism of fluoroquinolones. It inhibits topoisomerase 2 and 4, or it inhibits DNA gyrase. Why? If you inhibit those, you won't be able to uncoil the DNA. DNA will fracture and the bacteria would die because of DNA fractures. Okay. Fluoroquinolones. One thing you want to be careful about giving fluoroquinolones to elderly. What if you give fluoroquinolones to elderly patient and now they are complaining of some pain behind the ankle? What happened?
Cartilage damage.
Cartilage damage. Excellent. That's tendinitis. Okay. So because of the risk of tendinitis, we avoid giving fluoroquinolones in patients um, who have who are elderly to avoid tendinitis, and they also have some CNS effect. Okay, that's why we kind of avoid giving fluoroquinolones to.
Sir, one question.
Huh?
What is the function of topo 1 and topo 2?
Okay, so topo 1 is just going to cut one strand of the DNA and it will unwind. Topo 2 will cut both strands of the DNA and then it can uncoil.
Okay.
Okay, so if these are two strands, if it just cuts one, that's topoisomerase 1. If it just cuts both, that's topoisomerase 2.
Okay.
Okay, perfect. Now we have one disease where we form antibodies against topoisomerase. What is that? Anti-DNA topoisomerase. Anti-DNA topoisomerase.
Sir, systemic scleroderma.
Very good. Systemic sclerosis. Or scleroderma. Which is also having antibody which is called as SCL70, right?
70.
Wonderful. So we have um, systemic sclerosis, diffuse scleroderma. We have limited scleroderma and diffuse scleroderma. So this is systemic scleroderma, okay? Sclerosis. So we have anti-DNA topoisomerase antibodies in that, or also SCL70 antibodies. So that's about your DNA topoisomerase.
Sir,
So yes,
is it the same antibody helicase which I told you we found in dermatomyositis and polymyositis?
let me double check this.
Anti-MI2 is helicase.
Oh, really?
Yes, sir.
Oh, anti-MI2 is, and that's in dermatomyositis.
Yes, sir.
Let's see. Dermatomyositis. Did you already find it?
Yes, sir.
Okay. Where is dermatomyositis? I do remember we have anti-MI2. What were other antibodies in dermatomyositis? I always forget.
Anti-Jo1,
Uh-huh.
anti-SRP, and anti-MI2 is helicase.
And dermatomyositis comes in which chapter of USMLE?
MSK.
Oh, okay. I'm just searching. Why is it not showing up? Okay, I found it. Mm, you are right.
Sir, I sent you.
Okay, you sent me.
Yes, sir.
I'm just, wanting to write that thing in the whiteboard. So it's just one thing at just one place. So we have polymyositis and dermatomyositis. In polymyositis, we have what antibodies?
Uh, Anti-MI2, which is helicase, anti.
Oh, wow. And dermatomyositis has what antibodies?
I mean, it's common. We found
Oh,
in both.
okay. Yeah, Mi2s are helicase, man. And they also have, but scleroderma is different. This is
Yes, sir.
polymyositis and dermatomyositis. So
Yes,
we
sir.
have anti-SRP, anti-Jo, and anti-MI2, and in Helicase. Wow, that's wonderful. Okay, so we have one more correlation here in Helicase. So anti-MI2, which is also called as Helicase, we have that antibody in dermato and polymyositis. Okay, and apart from anti-MI2, we also have anti-Jo1 and anti-SRP antibodies in dermatomyositis. Very good. Thanks, Yashasvi. Okay,
No problem, sir.
so we got anti-topoisomerase correlation and anti-helicase correlation, and we also made anti-histone correlation from the DNA. Okay, so pretty much look at that. Pretty much autoimmune are targeting and anti-double-stranded DNA antibodies, which is in as what is that, SLE, right? So pretty much my antibodies are against the DNA, and you are having autoimmune disease. Okay, so that's your topoisomerase. Now this is the fun stuff. After we have separated the two strands, we have fixed the supercoils, it's time to make the DNA. Okay, but the DNA polymerase is very needy, meaning it has two requests to start transcript before starting the transcription. Request number one, it says I need hydroxyl group to start. Two, I will only replicate in 5' to 3' direction. Three, I need a primer. I need a primer, which is RNA, before I start replicating. These are the three conditions of DNA polymerase. If you meet this, then only your DNA polymerase is going to work. Okay. So we are trying to meet those conditions. Okay. And to meet those conditions, we have a wonderful enzyme which can bring RNA into DNA. Okay. And that's your next enzyme, which can bring RNA into DNA, enzyme number 6. And the name of that enzyme is
Primase.
primase. Wonderful. Now you are having primase which is trying to make RNA from DNA. So other name of this enzyme should be DNA dependent RNA polymerase. This is so high yield. DNA dependent RNA polymerase, that's the other name of primase. It utilizes the DNA strand and makes RNA. Okay, so that's your replication fork again. This is, let's say, your 5', this is your 3'. So this has to be 5', this has to be 3'. Now tell me, this is strand number A, this is strand number B. Where should I make leading strand if this is the direction of helicase? Where should be the leading strand?
Point
Point
A.
two.
Huh? A?
Yeah.
So we want the DNA replication from 5' to 3'. And in which direction the DNA will read? 3
3
dash
dash
to
to
5,
5 dash.
3 dash to 5 dash. So DNA will start reading, the DNA polymerase will start reading from here, 3 dash to 5 dash, 3 dash to 5 dash, 3 dash to 5 dash. So it's away from the helicase. So where, what it should be on this side, top,
Lagging.
lagging strand, right? So this is the lagging strand. And since you are reading from 3 dash to 5 dash and making from 5 dash to 3 dash, you can continuously make from 5 dash to 3 dash. This is going to be your leading strand. Okay, so I just reversed from the diagram of first aid. Okay, so make sure you pay attention to the labels. So this is 5 dash to 3 dash leading, and this is lagging. Now let's meet the requirements of DNA polymerase. Where should the primer be there? Okay, let's say this is your replication fork again. I'm writing 5 dash to 3 dash again, and this is 3 dash to 5 dash. Okay, now you want to make primer here first. Let's say this is your leading strand. I want to make leading strand here at point number B. This is A again. I want to make leading strand in point number B. You have to meet one requirement of DNA polymerase. You should give hydroxyl group to DNA polymerase 3. Okay. Now, the way by which I am saying, if this is your 3' end and I'm making RNA here with the help of enzyme primase, this end has to be 5', this end has to be 3', and whenever it is 3', it is hydroxyl group. Do you guys follow? This is 5' to 3'. I'm making primer. This primer is RNA. So this is your question. In which part of the DNA you will find RNA? And by reading that question, we all are like, what? RNA in DNA? This is primer. Okay. So if they ask you about where exactly you can find RNA in DNA, it's this primer. Okay. Primer will give 3' end to the DNA polymerase by giving hydroxyl group, and it will allow to multiply from 5' to 3'. Look, it has 3', so it can bring its phosphate. Now DNA polymerase can come and bind their phosphate here, and again from 5' to 3'. Okay, so look at this, it's replicating from 5' to 3' and reading 3' to 5'. Okay, so primase is fulfilling the requirements of DNA polymerase, but the problem comes here. Okay, any questions up till now? Okay. Okay. Problem comes here. You are reading from 3' to 5'. In order to fulfill the requirement, let's say we only opened up, we are gradually opening the replication fork, right? So initially only this much was opened, let's say. So you lay down one primer and then you make your DNA. Then this much opens up, then you lay down another primer and you make this much DNA. And again, this much opens up, you lay down another primer and you again make DNA. So you are going to make DNA in fragments in the lagging strand, and those fragments are called as Okazaki fragments. Okay. So since we are only reading from 3' to 5', every time you open up, let's say you opened up this much. So you again lay down primer here and then you replicate. You keep on opening, you lay down primer and then replicate. That's your lagging strand or Okazaki fragments. Okay. But you only need to lay down one primer in the leading strand and you can all the way go up till wherever the helicase ends. Okay. That's the function of your primer, which is also called as DNA dependent RNA polymerase. Okay. Now, since we have met the requirements of DNA polymerase, we are now actually doing the elongation with the help of DNA polymerase 3. Remember, this is DNA polymerase. So this is actually DNA dependent DNA polymerase. It is going to read the DNA and make another DNA. In contrast to primase, it was reading, look at this, is what it was reading DNA and making RNA. That's why it's called as DNA dependent RNA polymerase. But our traditional DNA polymerase 3 is DNA dependent DNA polymerase. These are so high yield stuff and words to know. Okay. Now, you can elongate your DNA with the help of DNA polymerase 3. I'm just going to make replication fork again. This is 5' to 3', 5' to 3'. Again, label A, label B. Where should be the leading strand?
B.
B, because it's going to read from 3' to 5'. Okay. And if I want to make the DNA polymerase work, what should I lay down here first?
5 to 3.
5 to 3. And what is that 5 to 3? What is this orange?
Primase.
Primer, right? With the help of enzyme primase, it's going to give hydroxyl group to the oncoming DNA polymerase. Now the DNA polymerase can come. This is your DNA polymerase. Okay, it's going to bring nucleotides and it's going to make chains of nucleotide. Okay, in this direction towards the helicase. Okay, so this is going to be your leading strand. So this is the point. 5' to 3' replicates, 5' to 3' replicates by reading 3 dash to 5 dash. Now this is very important. DNA polymerase is very, very specific. It will read from 3 dash to 5 dash and multiply 5 dash to 3 dash, 5 dash to 3 dash, 5 dash to 3 dash. Every time it moves from 5 dash to 3 dash, it will add nucleotides, nucleotide, nucleotide. But it also has one very special thing, is to look back. So if you are going from 5 dash to 3 dash, it's just going to look back to check if she made mistake. I'll not use word he. I'm kind of biased gender. No, whatever. If it made mistake, okay, I'll get in trouble sometimes. So if you are reading from 3 dash to 5 dash, making 5 dash to 3 dash, you replicate 5 dash to 3 dash, and you also have one 3 dash to 5 dash exonuclease activity. This is very important. So DNA polymerase 3 replicates from 5 dash to 3 dash, and it has also activity 3 dash to 5 dash exonuclease. Remember, this is exonuclease. Whenever you hear the word exonuclease, means it can only chop off the end nucleotides, end nucleotide, end nucleotide. It cannot nick in between. If it were be able to nick in between, that's called as endonuclease, not exonuclease. Exonuclease can only chop off from the ends. Endo can chop wherever it wants. Okay, this is extremely high yield. DNA polymerase 5 dash to 3 dash synthesis, and with the activity of 3 dash to 5 dash exonuclease, this is called as proof reading. Okay, do not afford to forget this. Okay, so this is your DNA polymerase 3. Now every time in lagging strand, you lay down primer, you lay down primer, you lay down primer, lay down primer, and every time DNA polymerase will come until it meets the upcoming primer. So you make intermittent sequence of DNA with RNA in between. There should be a way by which you can get rid of this RNA sequence, and we have an enzyme which can digest this primer. And what's the name of that enzyme? Enzyme number 8, who can chop off these RNA.
DNA polymerase 1.
Oh, yes, DNA polymerase 1 is going to chop off this primer, but in what sequence? In 5 dash to 3 dash exonuclease. Very important. So what's the difference between DNA polymerase 3 and 1? 3, they both are same. Huh?
Sir, what is mean exonuclease means like?
Okay, exonuclease. You have, let's say, this chain of nucleotide. If you want to cut this nucleotide from the end, you can use exonuclease outside. But if you want to cut right in the middle, you have to use an enzyme endonuclease. Okay.
Okay.
Perfect. Now, as I said, it has 5 dash to 3 dash exonuclease activity. So this is 5 dash, this is 3 dash. It can cut right from here. Tuck, 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 tuck. It will cut entire from 5 dash to 3 dash. Okay. Then later on, it can also synthesize from again 5 dash to 3 dash. It will cut 5 dash to 3 dash, again make 5 dash to 3 dash, and proofread 3 dash to 5 dash. Did you guys understand?
Yes,
Okay.
sir.
So what I'm saying, it will come, DNA polymerase 1 will come, it will knock off 5 dash to 3 dash, it will chop off. Okay, I've removed that. Now it can again read from 3 dash to 5 dash. So this is 3 dash, this is 5 dash. It will read from 3 dash to 5 dash and again make from 5 dash to 3 dash. So it's going to make here new DNA. And once it makes by from 5 dash to 3 dash, it can also do 3 dash to 5 dash exonuclease activity. So it's just like DNA polymerase 3, but with extra 5 dash to 3 dash exonuclease activity. So basically, DNA polymerase is 1. It can cut from both the side. Both the side, okay? 5 dash to 3 dash and 3 dash to 5 dash. In contrast to DNA polymerase 3 can only cut from 3 dash to 5 dash. Okay, it can only cut when it's back. DNA polymerase 1, whether it's here or here, it can cut on both sides. Okay, any questions?
So how can one, act on both sides?
So I mean, it's the property of one. I don't have a very good answer of how it can act, but it has
No,
chopper on. Yeah, go ahead.
sir, that brown one from 5 to 3.
Uh-huh.
So, in which direction it would going to 3 to 5?
So okay, this is 3 dash, right? This is 3 dash to 5 dash. This is the parent strand, and we formed a primer from 5 dash to 3 dash. This was the primer, right?
Okay.
Now we want to cut from 5 to 3 dash. So you will start from here and you will start cutting this primer from 5 dash to 3 dash. You had cut, and now this is your DNA polymerase 1 with the help of one cutter. So it has both cutters from 5 dash to 3 dash, and it has also other cutter from 3 dash to 5 dash, and it has also one glue by which you can add nucleotides. Okay, so with 5 dash to 3 dash cutter, it will cut first. Now it can add nucleotides here. Add nucleotides here. So this is a, let's say you cut here 5 dash to 3 dash, 5 dash to 3 dash, 5 dash to 3 dash. Is it okay? You cut here.
Yes.
Now this is 3 dash. This is again 5 dash. You can add nucleotides. You can again make a blue strand here. You add nucleotide, read 3 dash to 5 dash, synthesize 5 dash to 3 dash. So with the help of this activity, you can add nucleotide just like DNA polymerase 3. And then again, you can proofread. You just multiply from 5 dash to 3 dash, and again, you can proofread from 3 dash to 5 dash exonuclease. Did this answer your
Yes, sir.
question?
Yes, sir,
Okay.
got it.
So it's just cutting this primer and adding a DNA here. Same like DNA polymerase 3, but it has just one extra cutting function from 5 dash to 3 dash. Okay?
Yes, sir.
Wonderful. So DNA polymerase 1, and now you know. Now, after you added DNA here, after you added DNA here, you need one glue to attach these ends, right? It's pretty simple. So if you just need glues, and that glue which can join Okazaki fragments is called as DNA ligase. Pretty simple. Just attach it. That's your DNA ligase which joins the Okazaki fragments. That's also your question. So that's your DNA replication in summary. 9 steps. You have the origin of replication, data box. Open it up with the help of Helicase. We have deficiency of Helicase, you get Bloom syndrome. Keep that strand separated and prevent attack from nuclease with the help of single-stranded binder protein. You remove the supercoils with the help of topoisomerase. Irinotecan, topotecan are drug against topoisomerase 1. Etoposide, tinoposide are drug against topoisomerase 2. Fluoroquinolones can inhibit prokaryotic topoisomerase 2 and 4. You start adding primers. We have leading and lagging strand. We have fragments of lagging strand which are called as Okazaki fragments. Primase is an RNA in the DNA. Primase is also called as DNA dependent RNA polymerase. You keep on adding the DNA with the help of DNA polymerase 3, which synthesizes from 5' to 3', proofreads from 3' to 5'. You can remove the RNA primer with the help of DNA polymerase 1, who has exactly similar activity like DNA polymerase 3, and one new which is 5' to 3' exonuclease, and you join the Okazaki fragments with the help of DNA ligase. Okay, that was 1 minute summary of your replication. Any question? Okay, now one thing I just want to, that's the end part of the telomerase. Let's say 10th part. Okay, why do we need telomerase? You are replicating your DNA every DNA will have primer in its end. Meaning, let's say after replicating, the one end will always lag behind. What I mean to say is, if this is 5 dash to 3 dash, this is 5 dash to 3 dash. I said you read from 3 dash to 5 dash and you synthesize from 5 dash to 3 dash. So you will have to add primer here. You'll have to add multiple primers here. Okay. And I said DNA polymerase 1 can delete this primer and add a strand of nucleotide. But it can only add if you are having some attachment. So remember, this is 5 dash to 3 dash, right? So you are adding DNA from 5 dash to 3 dash fashion. So once you remove this, you should have again a 3 dash and here. I don't know if you guys are understanding this or no. We removed this primer, and if you want to add one more nucleotide here, this is already an hydroxyl group. So we have a hydroxyl group. So you can add here more DNA, right? But the problem comes here. Let's say this is the DNA you are making. You delete this primer. And remember, this is 5 dash to 3 dash. And I said DNA will only multiply for 5 dash to 3 dash, and it will need hydroxyl group. So if I delete this, who is going to give hydroxyl group here? Can anybody give hydroxyl group here? No. It will not. So this part of the DNA will not have hydroxyl group. So just a moment. So if that part, if this part is not having hydroxyl group, so every time this segment will shorten. Every time this segment will shorten. With each and every replication, this part of the strand will keep on shortening. Okay. So you have extra lengthy lagging 3 dash end. Okay. Be with me. So this is how your new DNA will look like. This is the 5 dash end. This is the 3 dash end. This end got shortened because you were not able to do anything here extraordinary because there was no hydroxyl group here. So this part will keep on getting shortened. So telomeres are the ends of chromosome. These are the telomeres. Oops, I draw a little hard there. These are the telomeres which are ends of the chromosome. With every replication, your ends of the chromosome will keep on getting shortened like this. To prevent shortening, we need the help of enzyme telomerase. What telomerase will do, it will bring a RNA template. So this is the RNA template. Every telomere will have a common sequence. Let's say the RNA template here is this is the template you need to remember, TTAGGG. TTAGGG. This is the RNA template. And this RNA template, luckily, will be almost complementary to the ends of the 3 dash here. So if I'm writing again GGG here, it has to be TTA. So you will find something complementary in the ends, right? Like this is CCC. Okay. So this is how the telomerase will bring the RNA template with it. Okay, it will attach to the 3 dash end because it's complementary. Now, giving the template of RNA, it will add more DNA to this end. So here it will add A, T, C, C, C here. So it will further elongate this end of the chromosome, right? And now again, another DNA polymerase can come and take this as complementary strand and again add new strand of the DNA here. So this is how it is going to prevent the shortening of the chromosome. So new DNA polymerase can come here, and this can add, and of course we want to do it from 5 dash to 3 dash. This is already 5 dash, right? So if you want to make something complementary, you have to add one primer here first, and then you can add here. Okay, this is how you can elongate your DNA. Did you guys understand or no?
Little.
Okay, so what I'm saying is, did you understand this part, that this is um this one, that 3 dash to 5 dash, you are just extending this region. Okay, let me just, okay, did you understand that why we have one end shortened?
Yes, sir.
Because we remove primer and there is no hydroxyl group, so we cannot multiply anything here, right? So let's say this is your 5 dash end, which is shortened, and this is 3 dash. So again 5 dash and 3 dash. Okay, you are going to have some common sequence here in the 3 dash end. Let's say we are bringing one telomerase which has sequence T T T G T T A G G G. Okay, T T A G G G. So this is going to stick to this end if it has C C C, right? This is going to stick, correct? Now this enzyme, which is telomerase, is reading RNA. By reading RNA, it can make a DNA strand here. So it can make AATCCC. Is that okay?
Sir, where it's come from? Like TTA, GGG, this
So
one, this term. Yeah.
TTAGGG is a sequence of telomerase. Wherever you see a sequence, just like 80, 80, 80, 80 Tata box, the sequence of the telomerase is TTAGGG. TTAGGG. If you see that sequence, that's the sequence of telomerase. It carries
Okay,
that
sir.
sequence with it. Okay, and
Uh-huh.
that sequence is actually complementary to the end part of the telomere of mostly all chromosomes. So it can just actually stick to the end part of the chromosome. Does that make sense?
Yes, sir.
Okay, now you are reading RNA and making DNA. So what kind of enzyme should you call this? RNA dependent?
RNA dependent, sorry, DNA polymerase.
RNA dependent DNA polymerase. So it's reading RNA and making DNA. So it is also called as reverse transcriptase, correct? It's reading RNA and making DNA, just like HIV. So it's reading RNA. This is the RNA template. Okay, this is RNA template, and it's making DNA out of it. So you read RNA and you make DNA. Can anybody catch what mistake I made? This TTAGGG is the sequence of the DNA. Okay, so RNA primer has to bring UU here instead of TT. So you just write UU. So that has to be RNA, right? So this is the sequence of the telomerase. Now, once you have done that, you also want to make a strand here, right? What's the fun in extending just this strand, which is 3 dash to 5 dash here? You also have to extend one strand here, which is complementary to this strand. So so you just remember, I can just extend this strand. This is 5 dash. I can multiply where, okay, you tell me, where should I read and multiply? If I want to somehow extend this strand, I want to add a strand here. Where should I add the primer? I want to read this strand. So it reads from
3 to 5.
3 to 5. So I'm reading from here. So I should add primer here, right?
Yes.
And I can multiply from 5 dash to 3 dash here. So this is 5 dash. So of course this has to be 3 dash. So now 5 dash is this one. So this is 5 dash to 3 dash. So you can make another DNA here. So you shortened this DNA. This shortened DNA can be elongated if you keep on elongating the other strand and make a complementary strand to it. If at all you don't remember, just remember telomerase is a very good enzyme which can prevent shortening of the telomeres. Okay, you don't need to know the exact mechanism, but you can elongate the shortened telomere with the help of telomerase. If you are not able, telomerase deficiency, if you have telomerase deficiency, your chromosomes will keep on shortening, and you have problems, right? Your chromosome degradation will be enhanced, your cells will not be able to multiply, and you're all in trouble because you cannot replicate. Okay. Which cells will have very high concentration of enzyme of telomerase because they want to rapidly multiply? So cancer cells will have very high concentrations of telomerase, right? So that's very high yield. Cancer cells and all the cells which are rapidly multiplying will have very high concentration of telomerase. Yeah, I think that's it about DNA replication. Any questions? Did you guys understand or no?
Yes,
Yes,
sir.
sir.
Wonderful. Okay. So let's stop here.